so in our previous two videos, we talked about this process right here, transcription, which takes place in the nucleus. And we talked about generating this mature mRNA. Of course, at this point, it's had, introns removed. It's had a GTP cap over here, and it's got a nice long poly A tail. And this thing's ready to be translated. So before we go any further, I'd like for you guys to think about what this word here means, translation. What does that word mean to you? It doesn't have to be biology related, just in general. What does translation mean? Okay. So you should have come up, if you Google it, you should get this answer right here, this basic answer. The process of translating words from one text or one language to another. Of course, that uses the word translate in it, but this is the relevant definition right here. You're taking things and putting them from one language to another language, right? So much like if I say hello and you say salud, or, hola, or one of those words, that's a translation, right? You're translating into a different language. So what languages are we talking about in this case? Well, we have our nucleic acid language, in which we are dealing with, to start with, in our DNA, adenine, guanine, cytosine, and thymine, or A, T, C, and G, right? Then we're transcribing or copying that into our RNA language, which is really the same language, right? You could say it's A, U, C, and G. It's a little bit different, right? Put my Oxford comma in there. So there's one little difference in letters in this U right here. But other than that, it's pretty much the same language, right? You could think of this as maybe, British English right over here, right? And we're copying it into, American English, if you'd like to think of it like that. But the main point to take away is that this language is really the same, right? We're not changing languages here. We're simply copying. Now, this next step we're going to do, translation, means we are going to an amino acid language. I have an amino acid chart over here with all 20 amino acids that are found in your body. And of course, if you'll remember, they have different property. And if you really remember, they have this carboxyl group here, COOH, this amino group over here, right? This NH2 amino group over here. That's from way back when. Each of them have these, right? Carboxyl and amino, right? Amino and carboxylic acid, amino acid. So we need to take it from A's and T's and C's and G's in our DNA, copy it to A's and U's and C's and G's, and then translate that into one of these 20 different amino acids, right? Of which these have 3-letter designators. If you'll notice this, you have HIS, LYS, ASP, GLU. You probably won't see this too much, but they also have 1-letter designators here. If you'll notice, R, H, K. These are the 1-letter designators for each amino acid. So that's kind of the way we speak in amino acid language, either with this 3-letter designator for the most part, and sometimes if you have a really long chain, you can use this 1-letter designator to indicate these different amino acids. Okay, so messenger RNA is ready to meet these other RNAs, which are going to be useful in this process. Of course, messenger RNA is involved in transcription and translation. These two over here are only really involved in translation. Ribosomal RNA, rRNA, makes up the ribosome. This is, if you'll remember from way back in the year, I told you RNA can form protein-like structures. Of course, there is some protein in here in these ribosomes, but a lot of it's made of RNA, right? This is a protein-like structure. This is an example of one of those structures that RNA can make. And ribosomal RNA, also known as, you might have guessed already, little r, RNA. Fix that little a right there. rRNA makes up the ribosome, and a ribosome has a 50S and a 30S subunit, a large and small subunit up here. That's why there's a blue and a yellow portion. But we'll watch that in action in a second. And you also have this thing called transfer RNA, or little t RNA. This is what physically brings those amino acids, right? This could be methionine, MET, or one of those types of amino acids. But this tRNA is what brings that amino acid to the ribosome and adds it to a growing chain. And I think as we go through this, you'll see kind of a little bit better how this whole thing works and breaks down. So the first step we have in this is initiation. This is where we have the 30S subunit and the 50S ribosomal subunit kind of connect. And this red strand right here, this is our mRNA molecule. mRNA molecule. It's just going to show this over and over again. This first step is called initiation. So we basically have this tRNA, which has what's called an anticodon on it. Anticodon are the 3-letter designators that are on a tRNA molecule. And it's going to attach to a complementary codon, such as AUG. Here, I'm going to draw up here since this thing just kind of repeats over here. AUG, if you'll notice, that's in RNA language, that's complementary to UAC. And this is on the, I'll draw the tRNA molecule up here, and it has a methionine. And this is my long string mRNA right here, blah, 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 blah. There's more bases. There's probably a, 5' GTP cap and a poly A tail, 3' poly A tail, right? So you probably have a non-transcribed region over here, but my point is you have complementary bases that line up on this anticodon. This here on the tRNA is called an anticodon. This here, that's on the mRNA molecule, and this is really one that you need to remember, is called a codon. A codon. This is very important because this is how we're going to decipher what amino acid goes where. So the next step in this process, right, we have these 3 sites, the E site, P site, A site. E site's the exit site. But for now, what we're going to have is that first amino acid, methionine, is going to be transferred into the next, right, the second codon here with this anticodon that comes and matches up with it. And this methionine gets transferred over. Now, what it doesn't show you on here is that this, you can think of it either way, either this mRNA moving through here and being read like a tape of some sort, or this ribosome moving across this mRNA, but you're going to have a movement here. You're going to have movement, right? This if you can imagine, this ribosome is going to move down in this direction. And this methionine tRNA in here, over here, is going to move over to this E site and eventually exit. And a growing chain is going to be coming off of here. And you're going to have a bunch of amino acids added to it. So the last step, termination. When you finally hit something called a stop codon, what's going to happen is this ribosome is going to disassemble for the time being, and it's going to release this finished polypeptide, right? This finished polypeptide. Now, that polypeptide might need to be further processed, somewhere else in the cell that might need to undergo further processing, further folding. But the point is that's going to end up folding into your complex protein-like structure somewhere on down the line with all those beautiful alpha coils and things like that. So that's pretty much it for translation. The next thing we're going to talk about is how we actually work with this code. Imagine, let's just review again. You have initiation, in which you have a tRNA that's bringing a single amino acid, typically methionine is your start codon. Ribosome assembles, and then you start slowly as this protein kind of elongates and moves through here. You start slowly adding to a growing chain. Eventually, you come to a stop codon, and let me get this going. And that protein is going to be released, that finished protein, which is located up here, right? This is the finished product. And that's it. That's it for translation so far. In the next video, I'm going to talk about actually working with codon charts. But that's going to be it for now.